and they divide some more, you get this mass of tissue. And then you can see within that mass of tissue, some of them have the X from the father, some of them the X from the mother being expressed. So you can see that throughout. That's why they refer to this as mosaic cells here, mosaic expression. So if we look at those disorders that are on the X chromosome, we're always going to have affected sons. The mothers could be carriers because they could have a good X and then X that's expressing, one's not. So they could be carriers, but the sons will always be expressing it because they only have one X. So if they have an affected, a mutation in a gene that's on the X chromosome, they don't have any opportunity to have the other allele that could be functional. So they're going to be express that. So females carry altered copies. They won't be affected. They also have a normal copy and expressed in some cells, and that's usually enough to have adequate cell function. And then there are 2 or more affected males in several generations. What we see in the pedigree, usually there's more than one male affected, and then we see them unaffected females. So we'll look at the pedigree, and then male-to-male transmission can't occur. So because males have to get a link that's a bit of Y to their sons. So female carriers have 1 in 4 risk of having a son because they have 1 in 2 chance of having a male, and they are carried. There's 1 in 2 chance that they'll be affected. So that means it's a 1 in 4 chance that they could have an affected son. If they're carrying a female child, there's a 1 in 2 risk she'll be a carrier. So if we look at pedigree, we see that carrier mothers, carrier females, I should say, that affected sons only, and carrier females throughout. We can have affected sons in every generation, and if no father, those fathers, sons, here are a few of the X-linked recessive conditions that we see that something that you might have heard of. Hemophilia, good one. Everybody's heard of that. Muscular dystrophy, everybody hears of that. And anyway, these are a few others that are common. Fragile X. This is a really well-developed pedigree here. And this is one for hemophilia. You can see the back way there. This is the royal family and European royal family, I should say. You can see because it stops here with here's Prince William and Henry down here. There's Diana Charles. But it goes all the way back. You see how this hemophilia was traced throughout. They ended up this branch here now. Carrier mother, carrier here, and then some carriers, but nonetheless no genes for hemophilia down through here. But it's interesting that you follow us back through here 7 generations. So it's difficult to get a pedigree that large unless you've had some family like this was keeping track of it for years and years. So unless if you're part of royalty, maybe you can get this kind of pedigree for your family. But this is a good example of how you can see all these different symbols used and see how it's been carried on and get track of this X-linked trait. Here's our best square to take a look at here. Mother and father, and we have here this is the normal, most common is having a carrier mother, carrier female, and a normal male. And then so we look at the odds there. So the sons are 50% normal, 50% affected. And we see the daughters same way, 50% normal, 50% affected here. So and here just using the 2 X's, X1 chromosome normal allele, X2 is the diseased allele. So we have these 2, and here's the normal for the father, diseased. So there's a chance there. This is another example here to show you that here, affected father and a homozygous unaffected mother. So now we have affected father. So again, he can only transmit that to his daughters. And so we'll only see it there. The sons are always going to be normal in this case because the sons will get an X from their mother. Those are both normal, so they're going to be okay. So the daughters will always have to have the trait because the father contributes 1 X to one of those X's, and it's always going to be an affected X. So we can see that an affected father, normal mother. X-linked dominant inheritance. We get these twice as common in females than in males. So that's what we look for, twice as common in females. We don't usually see skip generations because it's a dominant trait, so we're going to see every generation in somebody, except we don't have any father-to-son transmission again. And here it is males and females, every generation. So we can see that X-linked dominant. Here's another table I put together just to have to look at to help you distinguish between X-linked dominant and X-linked recessive, and to consider. But if you think about those, it kind of makes sense, but how we can't, the X-linked, how there's no father-son transmission. Remember, you always get one X from the father, one from the mother. If you're a female, you've got to get, whatever the father has, that's what you're going to end up with, one of them. And then you don't know whichever one of the two from the mother. So you can kind of make, these are a little bit easier to visualize, I think, than the autosomal genes, but these are, but nonetheless, this is just the same thing, put the table for you. Then there's the Y-linked genes. Now again, those are only coming from the father to the son. And so these are all co-android genes, exclusively from father to son. There aren't that many genes on the Y chromosome. If you remember back when we looked at pictures of all those chromosomes, the Y was really relatively small, very small actually, particularly compared to the X. So there's not much to the Y. There's primarily genes that are necessary for that embryo to develop. There's not, like I said, not a lot of extra genes there. There's some genes to differentiate, so they just keep these going. Transmission of Y-linked traits strictly from father to son. So it going to be father, son, father, son, father, son. No chance of any female. All the generations we keep seeing show up again. And this compares to the Y-linked inheritance pattern. Okay, so the last one, I had the sex link, we had autosomal, with the sex link, we have now this is what's called mitochondrial inheritance. If you remember the mitochondria from biology class, within a cell, they are another organelle, and those often referred to as the power plant of the cell, the energy station of the cell. And that's where we generate all of our energy. That's where we generate, our cells generate all that ATP that we need, the energy that we need. But within that mitochondria, they have another circular piece of DNA. It's a mitochondrial chromosome referred to. So this is separate from our other chromosomes. So in the nucleus we have those 23 pairs, but in the mitochondria we have these circular pieces of DNA. Now there can be more than one of those per mitochondria. And there can be more than one mitochondria per cell. So different cells have different numbers of mitochondria, and different mitochondria have different numbers of those little chromosomes. So the cells that are going to have more mitochondria are going to be the ones that require more energy. And we find that those cells that have to do with things like brain function, neurological function, cardiac tissue. So these are places which need a lot of energy, or we're going to be always producing ATP. We're going to have a lot of mitochondria. So when you're thinking a lot, when you're studying hard, you're reading, and you get exhausted, geez, all I did was study all night, why am I so tired? It's because you have used it. It takes an incredible amount of energy to keep your brain operating at a higher level like that when you're using it. So there's a lot of mitochondria. So when we see mutations in these mitochondrial genes, then quite often we see the phenotypes of disease we see are these neurological type of diseases. If you look at some of these that are mentioned here, these are all neurological diseases here. Here's a cardiomyopathy, because here we're having mitochondrial mutation. It's the cardiac tissue. We see that kind of expression. So it's where there's a lot of mitochondria, a lot of this mitochondrial DNA here occurring. Usually these genes, mitochondrial genes are maternally inherited. It's not 100% exclusive, but for the most, in general, that's the kind of, that's the statement we refer to, is that they're maternally inherited. There are instances of paternally inherited diseases as well, mitochondrial disease-related diseases also, but mostly it's maternally inherited. These are harder to identify with the pedigree. Mitochondrial disease pedigree looks like some of the dominant issue. We affect people every generation, male and female both. Difference, no male to male or male to female transmission. And females could be symptomless. So that's about the only difference, but it looks a lot like an autosomal dominant. It's very tricky to tell. Now, fortunately, you won't have to rely just, you won't have just a pedigree and have to figure out from that if that's your only clue. You're also going to have patient history. You're going to have the patient's symptoms in front of you as well. So you're going to look at the patient, you're going to have this, or look at the chart and have all this information. What kind of symptoms do they have? And you look at a pedigree, you look at the symptoms and say, this seems a lot like a heritable disease. Look at the pedigree and start thinking, okay, now how is this inherited? So you're going to be able to have some of those clues. And you say, well, that's a neurological disease. Maybe let's check the possibility. Does this pedigree look like a mitochondrial inheritance, or does it look like just a regular autosomal dominant type of inheritance? So you have clues to help you along the way. It's not going to just be thrown blindly to nothing. So you're going to have some knowledge of what the pathology looks like for certain diseases, and that's the thing you'll learn along the way, depending on where you happen to be working, what field you're working in. So you'll have that help. There's also this phenomenon called genomic imprinting, and we see that depending on what allele the gene has been inactivated on, we'll see different symptoms, different pathology, or different phenotypes being expressed here. So this is called gene silence. This is a form of gene silencing. It's called imprinting. Let me show you an example here. These two guys here, they both have, if you look at 15, and if this comes to its father, if this region is deleted, or it's not there, it's missing, then we see, but it's still there in the chromosome of the mother, then we see this symptoms occur, and it gives us what we call a Prader-Willi syndrome, identified by Prader-Willi. And you see this type of phenotype, there's some neurological developments and neurological delay development. There's birth issues, and you see the loss of swelling down, a lot of edema, and shorter stature. If this particular region, though, is deleted from the mother's chromosome, but still in the father, so we have expression from the father, then we see the same region, the exact same chromosome gene. Then we see a different Engelmann syndrome. We see just a little bit different type of phenotypic expression now. Again, some neurological problems here, development here. They have different gait, trouble walking, some joint issues, and some other problems with the joints and arms and legs. So we see these different phenotypes for the exact same genes being expressed, but it depends on whether it's from the mother or the father. So we call this again. This is called genomic imprinting, and this is just a really good example of that kind of phenomenon. So these kinds of things are, there's different factors. It's not like it'd be great if it was just really simple inheritance and follow along and figure it out and identify it that way, but it's always mitigating factors to change things. Okay, so that's essentially the idea, some of the basics and the real principles of the inheritance factors here. Any different questions about any of that? If we don't get all the way through a particular topic on Thursday, then I'll tell you, where we'll stop. Okay, I want to look at this. One of the things you remember, one of the things